this is kind of like a storytelling. I'm going to tell you kind of a little bit of the trajectory, so bear with me. So it's 2020, and by chance, I come across a paper that is called Rethinking Autism by Anne Donnellan. And I was at the time having a hard time publishing the research, that the results I had found, excessive noise and randomness in the motor code. And I flew to San Diego. She was the provost of the University of San Diego at the time, and gave me an appointment from her busy schedule. And we met, and I proposed that we do a research topic in the Frontiers platform on taking autism from a different perspective, from the movement perspective. And we gathered, she agreed to that. So we gathered 90 authors, and then we launched the topic. And I think that was not actually chance, because today we have well over 500,000 viewers and people interested all over the world. So I think that we did the right thing, and there was something there cooking that at the time was just a nascent thing, but then it took off. And later in 2015, I'm still in this mode of like trying to publish and not being able to get the results across in the research field of autism, and trying to have them see the significance of what this means for behavior and for everything that you're measuring or trying to measure. And I came across this paper in Science on precision medicine, proposing that we're at an inflection point in medicine where the incumbent curve it's all about data collection and description within disciplines, and the nascent curve taking off. We're poised to this very fast, rapid takeoff for precision medicine, meaning personalized medicine that combines all these layers of knowledge in the knowledge network and comes up with some kind of personalized profile of the person. It's been successful in cancer targeted treatment. So I thought, why don't I recast the problem of autism in the context of precision medicine? And perhaps people will see the importance of what I'm trying to do here. And so the idea is to take behaviors, which is the layer that poses the main barrier for me to do quantitative science, because it's all observational, or it was all observational at the time, and shift from a one-size-fits-all umbrella kind of blanket term, which is very heterogeneous and really hard to handle, and utilize the genomic information that we have and come up with a stratified autism that then enables us to do a precision model of autism with like a sort of fingerprint describing all these features at a precision level and the signatures of the person in terms of the behavior. But doing that not under the current deficit model that we have, which is just a laundry list of things that are wrong with the system, but rather do it in a way that gives us a chance to get at the readiness potential of the individual and harness really the capabilities and the predispositions of the nervous system that ultimately is combining all of this information and producing purposeful behavior, so that we completely come into a paradigm shift of what we're doing. And to do so, I wanted to leverage three contemporary revolutions: the neuroscience revolution, the genomics revolution, and the wearables biosensor revolution, which is severely subutilized in autism. And I come from a different field altogether, which very successfully is being using the wearables to basically digitize behavior. So to do that, I came up with this kind of branding idea of let's reformulate behavioral analysis into intelligent behavior analysis and shift from this vantage point of an external observer that is guessing and theorizing and sort of trying to infer what's happening in the brain and the mind and the body of that person, by combining it with wearables, sensors that actually read out the internal information in the nervous system, in the brain, and can get us to the mind, the mental spaces and the physical spaces. And so I decided that the time was right to actually combine this external observer with the internal observer using off-the-shelf technology that then would enable us to launch this at a scale and digitize behavior. Then the other thing to do was to come up with a compact set of tasks that would enable us to move this to the home environment, to the schools and to the places where life happens naturally. And so I took this old biomechanic pointing task that is very rich nonetheless and very difficult to model in robotics to get information about the voluntary states and the spontaneous states of the system along a taxonomy that I designed to study all the states, but in particular these two, because in a simple task like this, when you move forward to the target, you learn based on error correction, but then there is an incidental motion when you retract the hand very, very much beneath awareness that we could utilize to retrain the nervous system with minimal intervention, meaning not upsetting or stressing the system that would get upset otherwise by prompting and so on. And in addition to that, we could get this paradigm converted to a cognitive driven paradigm and establish different levels of cognitive loads and decision making and bring it to a completely different realm of cognition. And so there we would be bridging movement and cognition, movement and its sensations with cognition. And then we got this on the road and went to this is at a school nearby the campus where they train children with this applied behavior analysis paradigm, but they do it in a way that really robs the child from its autonomy, from its agency. So I said, let's do it with support first, but move towards a completely autonomous way of doing things and banking instead of external rewards, banking on the intrinsic rewards that the nervous system gets from being autonomous, from owning the action, from owning the consequences of the action and understanding that it is me doing this and I own this action. It is me in control here. So the children got their intrinsic reward. They became we gamified these activities and so forth. And in the meantime, we're digitizing everything that they're doing. So I realized that the handful of people that were using biosensors were following a rather old approach, which was assume a priori a theoretical distribution, take epochs of the signals that you're collecting with these biosensors, average them, and then come up with an assumed theoretical mean and standard deviation moments and so forth, but then losing all this gross data that for me was precious because that was like where the information most likely lied. So at the time that I was thinking about these things, I was doing a lot of computational neuroscience models and working with cortical spikes to which are basically digital spikes, and you do some transformation, get them into some kind of analogous continuous signal, and I was working in that field of spikes and so forth, and I thought, why don't we take the continuous stream of behavior and invert this model? And we start out with this continuous analog signal, turn it into a point process and come up with a digital spike. So we go the opposite direction. And in this way, we can study behavior naturally, but not just constrained to what the naked eye can capture, but go beyond the confines of the naked eye. So I did that, and then the same behavior then became a continuous stream that I would then transform into this thing that I call micromovement spikes. And instead of assuming a priori a theoretical distribution, I empirically estimated the family of distributions that most likely characterize all of the behaviors of this person and turn it into a kind of fingerprint or stochastic signature that let me look into the dynamics of that nervous system as the person developed over time. And then we launched a series of studies to actually test this paradigm. This was one of them by Dee Wu, and she connected all these levels of spikes. These dots here are the spikes, the peaks of the movements as we move to a target and then retract our motions And what we found was that there was an elevated noise and randomness in non-speaking individuals that began to acquire some pattern and periodicity all the way to the neurotypical speaking individuals. So we could actually model this and borrow all the computational neuroscience methodology to now study behavior in a different, completely different way. So this was a paradigm shift to the study of human behavior, of natural human behavior, and combined with the observation of behavior. So looking at the inside of the child. And indeed, the autistic system, this is the typical profile of the autistic system. I'm plotting here, superimposing the speed profiles of the forward motion, the touch on the screen, and then the backwards automated spontaneous retraction of the hand, which happens incidentally, and it doesn't really have an external goal or anything. But then you can see that both in the frequency and this temporal domain, there is a lot of noise, and that noise and that randomness impedes the creation of a predictive code by the system, because also this information serves as a reafferent feedback to the brain, and that's what the brain is receiving. And we characterized it statistically. It turns out this memory, this random distribution appear ubiquitously in all the biorhythms of the autistic individual. So they cannot feel at ease in this kind of uncertainty. So our job is to actually find ways to build accommodations to actually dampen this noise and help them understand the world better. So we did some math. We figure out essentially how to get targets for treatment and how to track the non-stationary nature of this code, and to study all sorts of behavior and digitize all sorts of clinical tests, including the ADOS, adapting other tests from other fields, and studying the sort of the progression, the maturation of the system. And so we did cross-sectional studies, we did longitudinal studies. This is an example of a cross-sectional study from ranging from 3 to college age, and seeing the transition in probability space of the different types of states that the body, how the body moves purposefully, and how this doesn't happen in autism, where again, we're looking for a target for treatment and published this in the Journal of Neurocomputation. And they did find that this maturation doesn't occur in ASD, but it is possible to shift it to this part of the space. So we found a noise to signal ratio as a target for treatment that we could shift, particularly utilizing the spontaneous variations in the movement and avoiding the prompting. And this gave us a way to begin to think of autism differently, defining a maturation path, defining a target for treatment, and revealing an age-dependent probability space that we could track across the lifespan. And then we decided, okay, so let's leverage the genomics layer. Now we kind of solved the behaviors layer. Let's find the genomics layer and start looking at this signature, and in a way connecting it to behavior through this taxonomy of the states of mental states and bodily states that ultimately depend on these three fundamental types of muscles: the cardiac for autonomic control, the smooth for involuntary, and the muscle skeletal for voluntary, which would give us the chance to connect to the proteins and the genes that produce those proteins in a systemic way, and study not just the brain, but study the entire system as it produces behavior. So we leveraged all these different databases of human genotype, tissue expression, of the SFARI database, and of the DISGENET, disease association to network, and OMIM, and came up with 20,923 genes from different diseases associated to different disorders, including neuropsychiatric and neurological and non-neurological, non-brain related disorders like the cancers and the autoimmune and so forth, and studied these 54 tissues and the expression of these genes on the tissues to begin to identify some of the tissue candidates that are most likely or highly, these genes highly expressed in autism, and that therefore might be most likely relevant to our quest. And so we use this parsimonious way of doing things, just looking at the gene expression, counting and binning, and studying in the context of a probability space using similarity metrics that would tell us the distance across these different disorders based on the expression in these 54 tissues, and then characterize autism and other disorders that go on and have a high intersection and perhaps go on to receive the diagnosis of autism or not, but also understanding the intersections, the shared space in the gene space for these 20,000 plus genes. And we realized that schizophrenia, ADHD, and Parkinson's disease as the three top genes shared, and also an autoimmune disease like lupus also has a high shared space. And then we also uncover genes and maximal expression in different areas that are shared across all these different neuropsychiatric and neurological disorders. And then went on and looked at this in a normalized distance metric space scale, and realized that indeed there is a continuum along the neuropsychiatric and neurological disorders, and then they're very different. And the difference here, the higher the difference, the more yellow or orange range of this color scale, with disorders like diabetes or cancers or autoimmune and so forth. And then zoomed in and realized that there is a continuum from neuropsychiatric disorders in the DSM and neurological disorders that are diagnosed by neurologists. And autism is right there in the middle, sharing a lot of these genes. So then it was important to look at the tissues most with the highest expression in these gene pool. And turns out the cerebellum and cerebral cortex are the highest expressed, followed by the frontal cortex and pituitary gland, and then regions that are fundamental for motor coordination, motor control, adaptation, self-regulation, and emotion. And so this gave us a sense of, like, now we know a little bit about stratifying autism based on these different overlapping disorders that share a common genetic pool. And so how do we detect this the earliest possible time? This turns out that neonates, there's a standard test, auditory brainstem evoke response test, that is quite early, within hours of being born. They give this test, and then they longitudinally track it for weeks later. And then we received a set from the New York State of data where we could look at these latencies in the different regions where this test is tracked. So they give clicks and bursts, and then there's a wave that propagates all the way to the primary auditory cortex, and it's standardized how they do this and how they look at this information. And so I thought, okay, there is a literature out there. Let's have a look at how what's happening here. And so with this in mind, we looked at about 70 babies, and 2/3 of them went on to receive a diagnosis of autism at around 3 years of age. So this is happening a few hours after birth. And already you can tell that the babies that went on to receive the diagnosis of autism along this pathway have a shift in their latency, so there's a delay. And not only that, there is a very narrow bandwidth of information that they are receiving, and this is cumulative, going through all this pathway of the 8th cranial nerve all the way to the primary auditory cortex, and inviting to think really seriously this as a fundamental basic block of everything else that happens with sensory processing, integration, and sensory motor transformations, because this delay and this narrow bandwidth is telling us that the system is not integrating the information in a way conducive of experiencing a percept the same way that somebody else is experiencing out there in the world. And so this would impede the type of social interactions, because it speaks to the sense of simultaneity that one needs to develop in order to align all the different temporal scales from different sensory modalities, ranging from microsecond to millisecond to second time scale. And if you don't have this at the basic sound level, where everything is sound, you know, including motion is also sound. So we can't really communicate with the world. So importantly, all these biosensors at this very early age enables us to follow these taxonomy that I'm proposing, with all these different muscle types, these three fundamentally different muscle types: cardiac, smooth, and skeletal, to track the levels of autonomy of the nervous system from the get-go. And frankly, we can do this at home and begin to understand how the system evolves from reflex to intent, to cognitive intentional acts, and how the system self-discovers intentionality, and bring this, bridge the motor to the cognitive level, and do this in a way that is comfortable to do it at home, and more recently in the lab, so we can detect it very early, and more recently in the lab, even empower the parents and work with the families, give them an app that then enables us to track the babies, and essentially use the micromovements approach combined with the ABR that they get anyways to understand the nervous system of that baby from an early stage. And so this is precision autism. It's leveraging the three major revolutions. And in essence, what I'm proposing now is go to the next frontier, and I call it the human kindness revolution, at an inflection point that I feel is happening with the community, that is bringing us from autism awareness, this would be our incumbent curve, to the nascent curve of actually having this autism acceptance, and shift the paradigm to have a research-based, community-driven approach that builds accommodations and support for the autistic individual, and doesn't see the autistic individual from an observant vantage point, but rather as a part of the human spectrum. So we should be redefining autism in the human spectrum, and this is what my research has been working toward. And I thank my lab and my many collaborators, and this is the end of my talk.
Jonathan, can I ask you something? Are you feeling like this was this is all a match in the way the kind of field is going at this field? I mean, the way in which people's lives are being explored and assisted and engaged with at this point. Yeah.
So, I mean, it's all heading towards, to some extent, potential for personalized evaluation and treatment, right? So at the genetic level, you can clearly see personalized factors that are unique to this subset of cases, are unique to this individual. And those point specifically to things that could be treated, particularly if you have genetic therapies that might specifically target that factor. And again, the same is, so if you're looking at phenotypic or neuroimaging traits in these individuals, you may be able to combine that information with the genetic data to get a better personalized assessment. So I would say that both in terms of neuroscience and genetics, that's where things are headed.
Yeah. So Liz, what kind of response have you got from, because you work with the community out there in UK?
Yeah, so I work with the community, and this is what the community wants. They want basically a paradigm shift where science looks at them as collaborators, where the individual is not seen as something broken that needs to be fixed or brought back to normalcy, but rather as someone who can flourish when given the support like everybody else. And so that's sort of the philosophy that I'm following in my research program. And I work with the families hand in hand. This is all informed by what they want. And so, you know, with COVID, it's been a challenge because all behavioral research virtually stopped. We couldn't go to the labs, the sensors, we had to reinvent, pivot and reinvent the whole thing. And the silver lining was that we were able to leverage off-the-shelf biosensors and send them home, and simple Apple Watches, cameras, and things that people can use at home. And so this empowered the families and made them into collaborators. So basically the families that collaborate with my lab and my center of excellence are my collaborators, like any other researcher. So they tell us what they need. And this is pretty much the philosophy in other fields, like in Parkinson, we work with the caregivers, in ALS, in the ataxias. It's really a different approach than what I've seen in autism as a field. So it's really where we're going with this.
Yeah, you actually mentioned to me, you sent me a paper and mentioned that a book was coming out by Alicia Broderick and Robin Roschino, is it?
Yeah.
Is it correctly? And the paper itself was called Autism Inc, the INC, the Autism Industrial Complex, which is raising some of the points that came up earlier in the discussions, which you're obviously very alert to. Could you speak to this a little bit, just to...
Yes. So in my role of this statewide center of excellence, and you can imagine a state where the prevalence of autism is at its highest in the US. All the problems that we have, the families, what they inform us is of these, they have been turned into a commodity. And there are several, it's an industry. Autism became an industry. It was branded according to Alicia and Rossignol, Broderick and Rossignol, as hope. First, there was the aspect of giving hope to the parents for a child that was normal, that we can bring your child to normalcy. And then here's the science that proves it. So trust us, we're doing the scientific thing. And what I found when I came to autism from the field of computational neuroscience was that the science was really weak. There was no real rigor on any of it. It was not reproducible. It was driven by a model that is not really peer review like it is in our field. A lot of conflicts of interest. The journals were a handful of journals that would not critique their own work. So it was a very different model than what we use in science. We criticize each other. We're open to criticism. It's open access. We talk to each other. We respect each other. I publish stuff. Here's the caveats. This is what I'm telling you. But here's the caveats. So we need to look at the caveats. This doesn't exist in the behaviorism world. It's really a very different type, and there's no waveform. It's all done by hands. And so this is what they call the science, and this is what they sell the families. And it's a multi-billion-dollar industry per year. And so the families caught up with it. It took a long time for them to, catch up with this, because it's not like, the way I look at it is like, if you're in engineering, right, and you build a bridge and you lie, the bridge, falls down and kills a lot of people. And immediately, this is a problem. So you can't lie. You can't afford to lie. But in this case, the lie and the misconception and the misdirection and the deception and so forth took a long time, because it took generations of people receiving these kinds of therapies to realize, oh my God, this is inhumane. This is conversion therapy, gay conversion therapy adapted to children. It's a shame. This is a Skinnerism. And the thing is that we're kind of experiencing in social media a little bit with all this fast reward and the social media, stealing all our data and doing with it all kinds of things that are, not unethical and so on. And so this is going on in autism. It's going on behind the scenes. The researchers don't really know about it because the researchers are actually impacted by this whole Autism Inc. As well, but they just don't know about it. They were just going with the flow. The only reason why I came across it because I started working with the community doing public service. And the community then told me, okay, this is what's going on. And so now I'm onto it for a number of years now, and working with people in economics and other areas, not necessarily neuroscience or genetics or anything like that, who can come up with a better model, a more humane model that actually helps us help the families as part of our human spectrum, not as something, you know, different from us.
Good, good. Jonathan, a comment on that? You finding the same thing?
The concerns of the autism community that it's just a transaction that happens and then we all go about our lives and it's not, and there's no real focus on helping people. I can see where that comes from because the way it's working right now is that we can really make a conclusion about an individual's autism genetics. In a small fraction of cases, can we say with high confidence that you have this disorder and that we can start exploring potential treatments. And so probably when in our interactions with families, in our communication with families, I think we could do a much better job of really explaining what is happening in those personalized medicine cases, which is where the focus is right now. So it's true that participation in a genetic study for probably 90% of individuals who enrolled in our study was a transaction that it began and ended in one day. And that was the end of it for most people. But that's simply a matter, it has to do with our knowledge and our ability to actually draw conclusions from the genome. So maybe it was only 10% of families where we could return it. So let me actually back up. In at least 10%, it might be closer to 15% of research subjects in our study, we returned a genetic result to those families directly, right? So we designed the study from the very outset to initiate that kind of an interaction with the family and with the primary care physician, and that we could initiate genetic counseling and then potential discussions regarding genetic therapies. But of course, the therapies themselves are really still basic science research that's going on. And so at this point, it's still very preliminary. So but still, that's probably not evident. It's probably not, it's not visible to the broader community. I think they really just see the initial transaction and that's it.
There was a question came in. I don't know if any of the three of you want to play with this one. Put in terms of a little case study, parents with very high educational attainment history and high IQ scores had a son in their 20s. The son has a severe autism expressed with motor difficulties, speech difficulties, severe attention deficit. As a high-level guesstimate, which factors would be most likely to be in play? Now, that's the kind of thing that you get presented with on a daily basis, Liz or Jonathan. I mean, what go ahead.
Yeah, you really wouldn't know until you actually sequence the genome, right? So it's not as if you can predict it in advance. But if you were to just try to say what the trends were, just because a family is young does not mean that an older father has a higher risk of having a new de novo mutation, but you still have almost, so it's like 1.2, 1.4 fold increased rate in the older fathers. You still have a pretty significant rate in young fathers as well. So the etiology doesn't have to be fundamentally different. So there could still be de novo mutations, and undoubtedly there will also be polygenic risk. So you really wouldn't be able to predict based on limited information what is the etiology in a single case until you actually sequence the genome.
At my end, what I tend to do is get kind of a sense of what the life of this kid is like day to day, what needs. And this is a question really for the engineers, the people that do build things that can help build accommodations for the child and make the life more comfortable and achieve the full potential. Because once you get this, these supports, then it's easier. It's just like anybody else, it's just, and so a lot of the parents, that's what they're asking for. How do I get my child to do X in an easier way? One of the things that is helping a lot is like the iPads, for communication. And so if we could develop, leverage the cognitive scientific revolution and develop ways to communicate better with support and so forth, that would be amazing for the school system. So this is sort of like my take on it. Not as much the part of genetic counseling or the genetic part, but more the practical day-to-day life that child. So there is like a disconnect. So we need to work together across multiple fields to kind of bring to them, okay, here's what they care about, and can we build it? And we have the conditions to do this. The technology and science has evolved to the point that we can do this.
Brian, could you speak to that from the sort of Neurocrine perspective about the Autism Inc. and the various things that Liz has been talking about? I've been really interested to hear this.
Well, certainly as a former adult epilepsy specialist before I joined Neurocrine earlier this year, when I hear about developmental delay, certainly because of my training and my former expertise, I always want to know about the past, and particularly whether there's anything suggestive of seizures. For instance, in CSWS, the disorder that we have a drug in right now, can certainly ultimately present with behavioral difficulties that can approximate an autism spectrum disorder. But what you often see in those instances that can separate it from other causes of autism spectrum disorder is that these children will develop normally earlier on and then suddenly much later in childhood have a regression. And oftentimes, although not exclusively, but oftentimes in the case of CSWS, can also have comorbid seizures. So certainly hearing about the young man that you mentioned in this case, would certainly want to know more about the development leading up to that, and particularly about whether or not there was anything suggestive of seizures. One of the other disorders that I'll talk about in my talk that is sort of on a spectrum with CSWS is Landau-Kleffner syndrome, LKS, where the deficits involve predominantly expressive and receptive language. And certainly it sounds like from the case that you presented, that was a very large issue that the caregivers were dealing with. I think, though, to go back to Dr. Torres's point, though, about this patient, what it brings out, especially if this patient has been suffering from autism for years and years, is just the absolute imperative need for early intervention and for these avenues for treatment to be available through schools and through other avenues, because the earlier, just like with epilepsy, the earlier that you can treat with anti-seizure medications, the earlier that you can intervene with behavioral interventions, the better. And so I would just stress once again, particularly for this young man, if these symptoms were present earlier on, is a fault of the system if this unfortunate man did not have interventions earlier that could have made a significant headway.
Do you, with the new discussions that are going on, the kind of things that we talked about earlier, do you find any of you feel yourself in an awkward situation about how you navigate between being an imposing savior, as it were, or a collaborator?
Well, I think it's, certainly we would like to view ourselves as collaborators with physicians. And certainly when it comes to the studies that we're doing, for instance, in CSWS, we can't do that without close collaboration, not only with the investigators and the physicians that are treating these patients, but with the patients themselves and with their caregivers. And we certainly understand that. And at Neurocrine, we've, try not to look at ourselves as savers or anything, but look as partners in this journey together. And really looking at children who have this condition where they have a high unmet need, where with CSWS that can result in autism-like behavior, there is no approved treatments, no approved treatment strategies, really partnering with with caregivers, with families, and with the investigators that are taking care of them to see whether, for instance, our medication can make a benefit, but also really having an interaction with patient advocacy groups. And that's something that I've just been so pleased about with Neurocrine when I've joined, is just that they have such tight connections with patient advocacy groups, not only for CSWS, but for other conditions that we're currently studying, and that we have medications that are under trials right now, where we really have tried to not view ourselves as separate, but as equal, and working together as a team to bring these, potentially groundbreaking therapies ultimately to patients.
Did you notice anything? One of the questions I've asked for the panel for later on in the day is what effect the pandemonium has had, in the sense that the whole notion of facial recognition of emotions, you're wearing a mask, everything changes. Solitude, is solitude good? Is solitude bad? Is that what something on the spectrum wants to do or doesn't want to do? Why are they playing more Minecraft? Minecraft is helping children with autism make new friends, according to reports and so on. Very interesting and completely surprising in some areas. What's been your experience of the?
Yeah, some of the surprises are that some of the kids are very comfortable at home, creating online presence and having friends online, somehow sheltered from bullying and other situations at the school. So we launched a survey and found this out, and one of the things we did at the center was create a horticulture program online that they would do planting and things like that, learn about seeds and plants and what have you. And this created social situations, very interesting social situations online. And so the pandemic actually wasn't as bad for the people that we were interacting with as it was for other people. But I don't know the whole answer to that. I mean, obviously we would have to have a very broad statewide poll to know. But in the cases that the families we've been interacting with, they actually feel comfortable at home, because there's a layer of bullying and rejection and imposition of certain therapies they don't want, that they don't have to do this at home. So it's a different type of interaction.